Good evening, everyone, and welcome to the Brain Products webinar on sleep research. This is the second time we'll be giving the webinar today, and we are happy to see such a great turnout of attendees tuning in from wherever you are in the world to learn more about our tools for sleep research. My name is Christina, and I'm a Strategic Product Manager at Brain Products, and I will be moderating this event today. I'm very pleased to announce that we will have presentations by two speakers, Eduardo Bellomo and Bastian Schrader, who I will introduce when I outline the proceedings of the webinar. One talk regarding the hardware setup for sleep studies. The second one concerns the handling of the data after you've recorded it. We'll give you a walkthrough of our manual sleep scoring solution in BrainVision Analyzer. Lastly, we'll follow up with a question and answer period. Our first presenter is Dr. Eduardo Bellomo, and he is a scientific consultant in our sales team. Eduardo's scientific background is multidisciplinary, drawing on experience in sports and physiology with a focus on EEG and additional peripheral electrophysiological sensors. Today, he will talk about EEG hardware and other relevant accessories that Brain Products provides for sleep researchers. Our second speaker, Dr. Bastian Schrader, is a scientific consultant in our support team. He was heavily involved in developing the updated sleep scoring solution presented to today, and he's an absolute expert with this. During his presentation, he will walk you through an example dataset and show you how to manually score sleep data within BrainVision Analyzer. As you can see, we have some great talks lined up today. So without further ado, I will hand the presentation over to Eduardo, who will surely deliver a fantastic and informative talk about hardware for sleep researchers.
Thank you, Christina. Welcome, everyone. You're going to spend the next around 60 minutes, and we're going to discuss about sleep research. So I have put together like an introduction that is going to go through the different aspects that might require our attention when we are planning to design an experiment and we are considering which hardware is needed for this application. So what is needed for sleep research? First of all, our participants are going to be sleeping simply. So we want to have a comfortable CAP. Whether the actual recording is taking place in a laboratory or at the home of the participant, we want the CAP to be designed in order to ensure the most natural sleep possible in order to make our results more valid and generalizable. We want to have also a long-term recording gel, so something that, contrary to the typical gel that is used for lab-based studies, doesn't dry out after a few hours. We want to have a device, an amplifier with an extended battery life. Usually, sleep studies might last between 8, 10, 12 hours, so we want to have the complete assurance that the amplifier is not going to turn off. And we might want to be able to combine the central measures, so the EEG, with a series of other sensors, of other peripheral physiology measures. And I'm going to navigate you through the different type of solutions that we have within the Brain Products portfolio for EEG, EMG, ECG, respiration, acceleration. And then I'm going to quickly mention some of the other sensors that we have, but are not so strictly relevant for this application. Finally, I'm going to spend some time adding some information for which might be relevant if you are planning to add a video recording to monitor the participants during the experiment, if you're planning to perform the study within the scanner environment in combination with fMRI, or if you're thinking more about something like a sleep at home paradigm and therefore you need a wearable amplifier. And then I'm going to hand over to Bastian for the exciting details on this sleep scoring solution within the analyzer environment. So as I was clicking through my presentation, you have seen a few images coming up and a few product names. And what I'm going to talk about, what is my section going to focus on, is a description of the actiCHamp Plus, several caps and electrodes and gel options that we can have when thinking about sleep and we want to use this type of amplifier. I'm going to discuss the sensors and the additional options. And again, Bastian is going to take care of the software part. So the star of my section is the actiCHamp Plus, which is a You can see like the image and this smiley lady on the right-hand side. Is an all-in-one lab amplifier, so it's able to record data, EEG data, thanks to the channel cards that are here on the top. From 32 all the way up to 160 channels, and also on the horizontal panel here has a series of aux ports, so inputs for what we call EXG signal, that is like an abbreviation for external type of signal or peripheral physiology. It's a powerful amplifier. A 24-bit amplifier can record the signal all the way up to 100 kilohertz and has a very wide bandwidth, which goes from DC all the way to 7,500 hertz. Very nice feature of the actiCHamp Plus is that it can work with both active and passive electrodes. And based on your preference, you can choose whichever solution you want. You might best fit your needs. Of course, the actiCHamp Plus is also compatible with two other types of electrode technologies, which are not that relevant for this specific application, so sponge-based electrode and dry electrodes. So I'm not going to spend too much time on them. The amplifier itself is pretty reliable. It has a wide operating range between plus and minus 400 millivolt for the EEG channels and around 5 volts for the out channels. So you don't have to worry about drifts or amplifier saturation, which might be a problem with other types of devices. On top of this, the amplifier is sitting on top of the battery. The battery is called power unit and can live up to 24 hours. So you can comfortably conduct your study without having to worry about, having the power to be turned off. So the battery duration is around 24 hours for a setup with 32 channels and 10,000 hertz sampling rate. Of course, the more channels, the higher the sampling rate, the battery life is reduced, but it's still, even with the highest configuration, so all the channels and the 100 kilohertz, you will be able to perform a recording still in the range of 10 hours, so you can count on it. Something that is pretty interesting of the actiCHamp Plus is that together it is equipped with a trigger out port. What does it mean? That anything that comes in through the trigger cable can be mirrored to another device, so it can be sent out in real time, and also has a sync out port, like a 3.5 millimeter jack, that can be used to sync with other devices. So you can easily integrate this type of amplifier with other equipment that you might have in your lab. In terms of electrode technology and caps, the first option you have is to use passive electrodes. Within the Brain product portfolio, this solution is called the BrainCAP with multitrodes, and the multitrode is the passive electrode itself. And the advantage of the multitrode, which is a classic ring electrode, is that it's a low-profile, lightweight, high-quality silver chloride electrode, which is therefore adapted to the head of the participant and is rather comfortable to wear, especially when leaning against a pillow or against a surface. Great feature of the BrainCAP is that it is fully customizable. We already have American Association for Sleep layouts in our portfolio, but again, you have the full freedom of customizing your caps. So you might want to have additional drop-down electrodes. For example, in this cap, I have two drop-down electrodes for the vertical EOG channel, and I have another one to measure some other sort of neck EMG, and then a longer cable to measure ECG. The actiCHamp Plus has a 40-pin connector for the EEG channels. So to use a passive electrode cap, you are either going to use passive cap with touch-proof connectors and a dedicated adapter, which I have here. So you would plug the 1.5 millimeter DIN connector in the dedicated inputs, and then with the 40-pin connector, you would plug into the amplifier. The other option is to use the classic BrainCAP connector, so this box, which has a 50-pin output. So you need to use a dedicated adapter that I have here, which is the 40 to 50-pin adapter. So on the one side, as the name says, accepts input from a 50-pin ribbon cable, and then you can use a 40-pin ribbon cable to connect to the actiCHamp itself. What kind of gel can we use for this type of electrode technology? So we have two options. For drop-down electrodes, both options are available at actiCAP, our long-lasting friend and cap manufacturer. So the first option is for drop-down electrodes, so some electrodes that will go on directly on the skin and not on the scalp, is the SAC2, is a toothpaste-like gel, which solidifies after, well, depending on the environment, 20 or 40 minutes, and ensures a perfect adherence and stability of these electrodes. For the cephalic electrodes, you want to use the Elefix, and actiCAP recommends to use to purchase the 180-gram tube, simply because it's very handy when it comes to actually filling in a syringe, so that you don't have to spend time with a dedicated spoon and so on. So you can fill in a 10-milliliter syringe and then apply the gel as easy as it looks. Since the Elefix is not abrasive, you probably want to find a strategy to degrease the skin before. So you can do this by wetting a cotton bud with alcohol and then gently moving the hair, or you can simply use the classic passive electrode gel, the Abralit, which has some abrasive power. So you would prepare the cap as you would for a traditional recording, and then you would add the Elefix in order to ensure cap stability. The other option is to use active electrodes. Within the Brain Products portfolio, these are called actiCAP. The active capital electrodes have been recently redesigned to have a very slim profile, hence the name. They are still lightweight and robust. The connector, the metal plate, is still in silver chloride, and they are active electrodes. So what does it mean? When we're thinking about active electrodes, it's probably easier to digest how the technology works if we first think about how passive electrode. So the passive electrode is placed somewhere on the body, usually if we are measuring EEG on the scalp, has an electrolyte that is used to improve the stability of the electrode itself and the signal conductance, picks up the signal and then sends it all the way to the amplifier. The problem is that the electrode and the cable sort of act as an antenna, so they can actually pick up, together with a signal of interest, they can pick up also noise coming from the mains, from other devices, from cable movement. And this might be problematic because it's sort of spoiling our signal of interest and hence reducing the signal to noise ratio. The active electrode has built-in circuitry that make happen a process that is called impedance conversion. The impedance conversion, you can also sort of think about it as a local preamplification, is picking up the signal measured at scalp level and then transferring it one-to-one to the amplifier, hence reducing the possibility for, reducing by a greater extent the possibility for additional sources of noise, again, like cable movement, also some noisy electrical environment, to spoil the signal while our signal of interest is traveling along the cable. For this reason, the signal quality with the actiCAP slim electrodes is outstanding, and this makes also cap preparation pretty fast. First of all, with this kind of electrodes, you get really good signal when with impedances that are in the range of 25 kilo-ohm. This is 5 times more compared to the usual standard of passive electrode, which is between 5 and 10 kilo-ohm. And on top of this, each of these electrodes is actually equipped with an LED, which changes color based on the impedance level. So when you start, all the electrodes are red, and then as you prepare the cap, squeeze the gel, move the hair, the color changes, hence giving you an intuitive and pretty entertaining optical feedback on the impedance level, which is something that might accelerate the learning curve of your lab assistants or graduate students, and at the same time is actually simplifying your job because you don't have to sit all the way and every time walking all the way to the recording computer to see the impedances level. On top of this, the actiCAP, the slim electrodes can be arranged on a snap cap. The snap cap is so called because it has a series of holders, which are called snap holders, which can keep the electrode in place. So now, the great thing is that if you want to mimic what can be done with a passive cap, you can simply remove one of the electrodes and then place it somewhere on the body. Again, if you have 64 channels, you have more possibility of sacrificing some of the electrodes, but again, you have the possibility of mixing and matching the electrodes in order to have a combined EEG and peripheral physiology solution. The gel that should be used for sleep application and active electrodes is again available at actiCHamp and is a sort of electrolyte cream called V20. It's kind of difficult to describe what it is, besides the fact that it feels really like a moisturizing cream. And the crucial thing is that it doesn't dry out. Usually the active electrodes, our active electrodes are used with a gel called Super Visc, which depending on the environmental conditions, so the humidity, the dryness of the room, might dry out after 4 to 6 hours. This V20 electrolyte cream doesn't dry out. So again, you can be sure that you are going to have a good quality signal throughout your recording, 8, 10 hours, 12 hours, depending on how much your participant is going to sleep. And of course, I have here an example of a 20 milliliter syringe, but again, you can also get bigger dispensers than populated syringe yourself. Sensors. So I've been sort of anticipating this part. You can have a solution whereby you use drop-down electrodes that come from the cap, and then they are applied with a dedicated gel and washers to the skin. And you can measure EOG, EMG from the face and the neck, or ECG. And this is possible both with the brain cap and with the actiCAP when we unsnap one of the electrodes. Here again, I have one example layout. This is a 32-channel brain cap for sleep recording, which is whose layout is in accordance with the American Association for Sleep Medicine. And similarly, you can achieve a very similar configuration with the actiCAP electrodes. Of course, you will need to re-reference the data in order to interpret them meaningfully once you are analyzing the data. The other option is to use an adapter, which will plug into the aux ports of the actiCHamp, and I have it here. This black box, which has an input for 3 electrodes. The electrodes are simple multi-trodes with touch-proof connector. 2 electrodes actually measure the signal as a reference to each other, and one of the electrodes acts as a ground. The good thing is that if you're using multiple big 2 auxes, you will need only 1 ground electrode because the ground is internally connected. So 3 electrodes for the first beep, 2 out, and then 2 and 2 and 2. And this is how it looks in a nicer picture compared to what I have here on my table, which is a bit messy. For respiration, we have a respiration belt, which can be used for non-intrusive respiration monitoring, so cannot be used for spirometry, and consists of an elastic belt, which I have here, a pouch with a pneumatic sensor and a transducer, and then here the actual sensor that then sends the signal to the amplifier itself. You can usually use it to monitor thoracic movements or abdominal respiratory movements, and the transducer converts the signal, the inflow into a positive going signal, the outflow in a negative going signal, and then this is actually used as a relative measure of change of respiration rate or inspiration-expiration ratio over time in arbitrary units. For acceleration, we have a 3D acceleration sensor, which is also available as a one-dimensional acceleration sensor. It consists of a small differential capacitor. This is pretty tiny. Oops, sorry. It can be placed anywhere on the body. It has within the sensor itself, there are 2 fixed plates fixed to a flexible silicon structure, which are sensitive to movement and output a signal that can be measured in G-force, so 9.81 meters over square second. So if you are using the 3-dimensional acceleration sensor, this is going to take 3 aux ports, one for the x-axis, one for the y, and one for the z-axis. This can be placed, for example, on a limb to actually monitor limb movement. In the REM phase, we have a total muscular atonia, so this type of signal might be useful to actually do a properly more informed sleep scoring, but can also be used eventually to monitor breathing. You can place it on the chest, on the abdomen, or to measure blood pulse, depending on how creative you can get with your solution. And we have a dedicated page that spends more time in providing you with a theoretical background on sensors, as well as some information on how to perform the recording, as well as how to analyze the signal on our website. Together with these sensors that are particularly relevant for sleep applications, we also have a GSR sensor, a temperature sensor, a pulse sensor, and if you think about including these into your paradigm, you can simply drop us an email or contact your local distributors, and he or she can give you more, or us can give you more information on it. So as a partial wrap up, we have seen what kind of cap, what kind of options we have in terms of cap, recording gel, electrodes. We are sure that with the actiCHamp Plus, we are in a safe position, in a safe spot, because the battery will not betray us. And we have seen what kind of solution we can have if we want to measure peripheral physiology. Now I'm going to spend a bit of time, just a couple of slides, discussing the optional and specific additional applications that you might want to consider. So if you need to also monitor your participant during the experiment, we have a dedicated add-on for the acquisition software called a video module or BrainVision recorder. And this is optimized to work for a network video camera that we provide you with. It's powered over the internet camera, so it both sends the signal and it is powered by this Ethernet connection. It's a full HD camera, has integrated microphone, night view, and the crucial part is that it is synchronized with EEG, and the synchronization is in the range between plus and minus 50 milliseconds, so it should be used only for monitoring and not for trigger derivation. The signal recorded with the camera, the BrainVision recorder video module and camera can be easily and seamlessly imported into Analyzer. There is, again, an add-on module for BrainVision Analyzer called intuitively video module, and there you can perform your analysis, and you can, for example, make a judgment call on whether to score a specific segment as a particular stage, or to highlight another portion of the data as an artifact because the participant moved. If you need fMRI compatibility because you want to do EEG fMRI sleep studies, you will need a different type of amplifier. So we have within the Brain product portfolio, our solution for EEG fMRI is the BrainAmp MR. It can come in a standard version and a plus version, and is a monopolar amplifier. So you will be able to use this type of cap with drop-down electrodes, but again, you will need offline re-referencing if you want to derive some sort of physiological channels or bipolar channels, and uses passive electrodes. For the MR application, the cap is additionally equipped with resistors, and the cables are organized in a neat way to prevent loops, and also the cable tree is shorter. So a series of features that ensure the best signal quality alongside the best safety. If you want to also use the sensors within the MR environment, you can combine the BrainAmp MR with another box, another amplifier, which is called BrainAmp EXG MR, and can measure. It's coupled with a box called aux box that you can see in the image. Is the one with the blue connector and the black connectors. So it has a series. It has 8 bipolar channels, so you can use touch-proof electrodes. And you can plug 3 types of sensors, so the acceleration, the respiration, and the GSR. If you need a wearable solution, then again, this might be these paradigms are starting to become more popular in the recent years. These kind of like sleep at home paradigms, where you start the recording in the lab, and then the participant goes home and sleeps in his or her own bed, and then comes back to the laboratory the day after. You might want a wearable solution, and the LiveAmp, which I have here, is what we would recommend. It's a very small amplifier, is as big as a credit card and pretty thin, very lightweight, so it can be worn potentially anywhere. You can place it on the cap, or like wear it on a dedicated pouch. And again, it's a wireless amplifier, so it can stream the data via Bluetooth, but this is crucial for sleep applications, for sleep at home paradigms. It can measure the signal directly on a SD card, so you don't lose any data, and you don't need to actually have a recording computer close by. It has a built-in lithium battery, which has a duration of around 4 hours, but the battery life itself can be further extended if we simply combine the amplifier with a USB power bank, the same you would use to actually extend the battery life of your smartphone. Yes, another feature is that the LiveAmp can be used with both active and passive electrodes. So the considerations that we have discussed in the previous slides are still holding for the LiveAmp itself. Some sleep-friendly features are the fact that the LiveAmp can use a flexible referencing scheme. So on the one hand, a LiveAmp 32 can measure 32 EEG channels in monopolar, or well, channels that are also drop down, or eventually, if you have a dedicated cap, you can feed 2 inputs and directly process it as a bipolar channel. So you will, in essence, you extend, although it's a 32 channel amplifier, you extend the range of inputs that it can actually take. It has a built-in accelerometer, which again is a pretty interesting feature to have when it comes to sleep research. And if you need to have additional sensors, you can combine it with the sensory trigger extension. You connect the two together, you connect the sensory trigger extension to a battery because it needs to be battery powered, and then you can plug your sensors in one of the 8 aux ports. Eventually, there is also the possibility of sending complex 8-bit triggers, but this is not very relevant for sleep applications. So we have also checked this box, and it's now the time for me to hand over to Bastian.
All right. So thanks again for this nice introduction overview about the hardware solution that we have for sleep research. In the second part, I would like to walk you through the software application that we can offer for the recording of the sleep data. So I will use the BrainVision Analyzer during my demonstration. So for those of you who know the software already, you'll be familiar with some of the steps that I'm going to use. For those who don't, I hope that I can also show you a little bit around the how the software works and how we can use it for the processing of EEG data. So the analyzer is a fully featured signal processing software. It can perform all necessary processing steps that are actually relevant for any EEG data analysis, be it in the context of sleep research or any other EEG paradigm. So it features many methods and functions for the pre-processing, for the data extraction, and also for higher level analysis that you would then use for to extract the knowledge that you want to extract from your data, be it a time frequency or frequency analysis, or even connectivity analysis, for example. It is self-documenting, so you have the entire pipeline at a glance due to the history concept that I will also show in a second. And it's also quite easy to use due to that. You can apply a batch process, so you apply the one pipeline that you constructed once to many data sets or many subjects at once quite easily. And it's compatible with other software and also other data formats. So it has reader for more than 50 data formats. Thus, even if you don't use Brain Products hardware, it's likely that the software can also read your data. It has also an integration with MATLAB to enable scripting, where it's not necessary in any other feature of Analyzer. And it's also compatible with other software, like, for example, the statistical software offered by BESA. For sleep research, it's important that we can, we have this rather specific situation with the data that we're processing. So these is multimodal data. We have different sensors at different parts of the body, and the signals that we record do have quite different characteristics. Therefore, it's important that we can process each channel individually and adequately. For example, pre-processing steps might have different parameters for EEG channels or for ECG channels. And this is possible in Analyzer as well. Even more specific requirement on the analysis part in the context of sleep research is, of course, that we can make use of the sleep scores in the data. So be it for the calculation of sleep parameters or to extract the data where a subject has spent time in a certain sleep stage. And for the process of the manual scoring, so the assignment of the scores to the data, it's also important that we can customize the display. So to hide specific channels, only show a subset of channels, and also to change the scaling in time and amplitude. All that is possible in the standard installation of the BrainVision Analyzer. However, the tool to actually insert, inspect, and work with sleep scores and also calculate sleep parameters from them comes with an extension to the standard installation. And those extensions in Analyzer we call solutions. They are free of charge. And for sleep, we have the sleep scoring solution. So the sleep scoring solution is designed is dedicated for manual sleep scoring. So that means it provides some navigational and also display options that are designed to help the scoring process itself. It operates along sleep stage markers, That means that it does not only insert a marker, so a marker in Analyzer is an annotation in the data set. It inserts sleep stage markers that indicate the beginning of a scored interval. It also allows to import scores from a different software. As long as you can export the scores from a different software and then convert them to BrainVision markers of this type, the sleep scoring solution can read them, and you can then go through the scoring and adjust it if you want. And the conversion from let's say a list of scores to a BrainVision marker file is not very complicated. Based on those scores, the solution can create a hypnogram, so a full night overview of the scoring, and also a sleep report reporting basic and common sleep parameters as recommended by the American Association of Sleep Medicine. The hypnogram is actually created by Microsoft Excel. So for this creation, you need Microsoft Excel to be installed. For the sleep report, you can export the report in a text file that you can open with any text editor, or also in a spreadsheet, and then you also need Microsoft Excel for it. Hallmarks of sleep, like the sleep onset and the onset of sleep cycles, are calculated in the report and also exported as markers again, as sleep report markers. And based on those markers, you can then perform any type of EEG analysis, higher level analysis that, well, is targeting your research question. And a very small example of that I would like to show you now in Analyzer. So let me switch to the software. Right, so for those of you who don't know the software, we have here at the top a ribbon menu with a number of menu items. For most importantly, we have here the transformation. So this gives you the overview of all the functions and methods that you can apply to the data. And this is extended by the solutions, as I mentioned. So once you have installed the solutions, you'll have them available as well. And here we have also the sleep scoring solution. In the primary, there is this window here that looks a little bit like a Windows Explorer. We see 3 data sets. So this is sleep data that's provided by the sleep laboratory by Björn Rasch. And we have 3 data sets here. So the workspace is basically a set of directories on your hard drive where you place the raw data into, and then you specify in your workspace file where Analyzer can find those files. I can activate those history files by double-clicking on them, and then they will extend to show you a child node. And this child node, the first one is called raw data, so it just gives you access to the raw data. And as you can also activate by double-clicking, and then you can see the actual recording. So we have EEG channels, we have EOG here, we have EMG, we have ECG, a respiratory response, and also 2 mastoid channels for re-referencing. Once you apply any of the transformations or solutions, it will create a child node towards the currently active one. And I prepared already a little processing pipeline, and I can expand all of those child nodes corresponding to the different transformations that I've applied. So I did 2 types of re-referencing with 2 different sets of electrodes. I adjusted the display by deselecting a couple of channels. Basically, this is just getting rid of the mastoid channels. And then I'm applying here a filter node. So let's just have a look at the transformation itself, the filtering. So in Analyzer, we have IAR Butterworth filters. And here you can specify the frequency settings. So the settings for the high pass and for the low pass filter. And then here's a display of the frequency response of the filter. You have the option to apply a notch filter if you have a lot of line noise in your data. And most importantly, for sleep research or for any multimodal research, we have here a table where you can specify the individual filter settings. So for example, here the EMG channels have a different setting than the EEG channel, than the EMG channel. Right, so this is the specification for the filters. I've already ran the transformation, so I don't need to do it again. So I cancel that here. And I can basically double click that node to get my filtered data. Here in the navigation bar, I can also just use these arrows here to scroll through the data and inspect it. Right, so based on such data set, I would like to demonstrate now how the sleep scoring solution works. So I can raise it here from the solution ribbon menu. And you see that this little GUI, this interface pops up, and simultaneously in the background, an interval of data is highlighted or is selected in that sense. If I scroll through the data by using these arrow buttons here, it will always jump to the next scoring interval. So it navigates through the data in multiples of the scoring interval. It always selects those. So these are 30 seconds of data. You can specify this to be a different length. And you can then use the buttons here in the sleep stage history pane to score the data. So by just clicking on those buttons, of course. You can navigate through the data and also assign scores. Also, basically you can use any of those buttons also from the keyboard. So while performing manual scoring, you don't even have to lift your hands from the keyboard, so you don't need to use the mouse for that. So when I score, you've noticed that the interval always jumps to the next position, to the next interval, and this is dependent on these auto advanced options. So if I untick those, go a little bit back, then you see that I can score the same interval, rescore it, so to say, in the same interval. If I tick the auto advanced option, it always jumps to the next position. If I tick the unscored only option as well, it always jumps to the next open non-scored interval. So this is important for the manual scoring process. Right, I can also use some two transient views to support my scoring decision. Transient views are views that are generally available within the environment of Analyzer. They are these detachable windows, and they will always represent the currently selected data. And we have here selected the complete scoring interval, so it really shows data from these 30 seconds. Let me just adjust the views a little bit to show only the EEG data. Here we can adjust the amplitude and the frequency range. And if I now raise my solution again and start scoring, you'll see that the views here update to the current scoring interval. So this can really support the decision of the scoring. Right, so for the mapping view, it's really not very informative since I only have 3 scalp electrodes here. Right, so I don't really want to score 8 hours of sleep now, so I just click OK for now with the solution. And then you see that I have a new node created here. And if I go to the markers, so this is displaying the markers that are in the data set, we can see that I have a number of sleep stage markers in the data set. You can also see them here in the bottom on the navigation bar. Right. So what I did before this webinar, of course, is preparing a complete scoring of this data set. And I converted those also to BrainVision markers of the correct type. I will import those now with a transformation called import markers, which basically reads such a file and then imports them to the data set. So this file here. And once that's done, we can see that in the navigation bar, there are a lot of markers now. So these correspond always all to a specific scoring interval. And if I raise now the solution based on this data set, it will tell me that it detects the sleep stage markers and will also load them into its scoring history. And you can see them here that I have already scores loaded. So as you can see here in the navigation bar, but also in these unrated left-right fields, there are 6 gaps in the scoring. So the scorer or the software, whoever scored this data set before, has left 6 fields unrated. And I can use the unscored only option, but I can also use the arrow with this question mark to jump to those positions directly. And then continue, for example, the scoring with the keys to go through each gap and perform some scoring to complete the scores. So I'm just scoring it arbitrarily now. Now we have 0 unrated scores in our 8 hours of sleep. So from this, I can raise the hypnogram or open the hypnogram. As I said, this will transfer the scores to Microsoft Excel, and Excel will use its plotting functions to create the graphic. The file itself is stored in the export folder of the workspace. So that's a directory on the hard drive that you specify when you create the workspace in the beginning. And it will also open up the sheet directly, as we will see in a few seconds. There you go. So this is how the hypnogram looks like. It has the full night overview of the scores of the stages. In orange are the REM phases, so you can also see the cycles quite nicely. Right. From the complete scoring, you can also create the report with the report button. It will also use, in my configuration here, it will also use Microsoft Excel and create a spreadsheet. I think it's a more readable solution, and saves the spreadsheet in my export folder. Just let's look at this. So we have it here. Here's the Excel sheet for the hypnogram, and here's the report. Let's just open it to look at it. So that is how the report looks like. It has quite common parameters that are recommended by the American Association of Sleep Medicine, and it has a more detailed overview of the stages and also the sleep cycles that are detected. It also saves the scores itself as a numeric value and also as a label, and it has also a marker template. So this spreadsheet is already readable by Analyzer, so you can import it into Analyzer, and it consists of these sleep report markers that indicate, for example, the sleep onset or the onset of the different sleep cycles. This sheet is also automatically saved as a separate file with the extension .markers, and we can import this file into Analyzer. So let's just quickly close the solution. So it will rewrite the markers, and then we can import the report markers. Just browse the right location. This is our export folder, and here's the markers file. Okay. So once I have the markers loaded, I can check again in my overview here. So I have my report markers and also a number of sleep stage markers. So based on these markers now, I can perform any type of EEG analysis, and I'm using these markers to extract the data of interest. I prepared already a little pipeline in a so-called history template. So this is another concept in the framework of Analyzer. It's basically a set of instructions for a data analysis pipeline in this tree form. So you have different nodes that represent, again, a specific transformation. And you can apply this tree to multiple subjects automatically, so in a batch process if you want, but you can also just drag and drop this node onto the data set that I want to look at. And then it will apply the steps in the sequence that I've specified here. So let me just explain what it does. It has 3 subsequent segmentation steps. Segmentation is the process of extracting a specific interval of the data. And I'm using the sleep report markers in the first segmentation to cut out only the first 3 sleep cycles. And in the second segmentation step, from these 3 sleep cycles, I'm extracting only the REM episodes. And again, the resulting data is dissected into 8 seconds. That corresponds with the sampling rate to 4,096 data points, which is optimized for a fast Fourier transformation. So based on these smaller segments, I'm applying the fast Fourier transformation, which transfers the data into the frequency domain. So these are frequency spectra of the REM data in the first 3 sleep cycles. And I have here 297 segments. And then lastly, I'm creating an average node, which basically average across all the segments to give me the average REM power for these 3 sleep cycles. So let's just look at the frequency spectrum of only the EEG channels. Again, adjusting the view a little bit so we can inspect it a little bit better. So here, not a lot of, there's not a lot to see, but I also created 2 more branches in this processing pipeline, and they start as a child node to the segmentation of the sleep cycles. So again, I'm working on the first 3 sleep cycles, and then I'm extracting, instead of REM intervals, I'm extracting N2 intervals, and here N3. And the rest of the pipeline is exactly the same. So in the end, I'm receiving average power, for example, here of the N2 stages. And I can simply drag the node into the data view, which will create such an overlay. So in the filled area here is now the REM frequency response, and the red line is the frequency spectrum from N2 stages. And we can see here a theta peak and also something that might resemble spindle activity. We can do the same with the N3 power, and this will of course create a much larger response in the data range, as we can see here. Right, so this is what I wanted to show as a very short example pipeline, how you can use the solution to create the markers and then use those markers for subsequent analysis. Let me just wrap up in the slides. So I hope that I could show that the way how you use transformations in Analyzer is quite an easy way to process your data. You have a very nice overview of what you did and how you can of your processing pipeline. And we have a large collection of transformations that cover basically all most of the common processing methods that you would need in an EEG data analysis. And together with the sleep scoring solution, you can also then perform a sleep-informed data analysis. The analyzer comes also together with a free scientific support. So that is us. If you have any questions to the software, how to use it, or how to implement maybe a specific analysis strategy that you have in mind, you can drop us an email and we're happy to help if we can. The solution itself, the sleep scoring solution, you cannot download from our website, but currently you will also you will have to write us an email and then just indicate that you are interested in that solution, and we can send it out to you. We have also more educational resources on our website. There are support tips and tutorials, specifically also for Analyzer. And we have this Go to Stage channel where we keep the recordings of webinars like this one, but also some that are more scientifically focused with a bit more of theoretical background that explains the algorithmic and the theoretical concepts that are behind the analysis methods that are implemented in Analyzer. Right, and with this, I would like to go back to Christina for the Q&A questions.
Thank you, Eduardo and Bastian. Those were really excellent talks. The first question for Bastian. Somebody is asking if they can display heart rate variability from an ECG during scoring.
We don't have a dedicated view that would display heart rate variability, which is often also important for the decision of a certain stage. But what you can do is to detect the peaks on an ECG channel. We have a certain solution for that as well. And then from those peaks, you can convert this into the heart rate itself. And then you'd have a new channel in the data set that displays the fluctuation of the heart rate in your scoring interval, for example, and then the variability had to be extracted visually from that heart rate channel. So that's as close as it gets to heart rate variability. So that would be our solution for it.
Okay, thank you for the really informative answer. I also have another question about solutions in Analyzer. If we have an automatic sleep scoring solution.
Yeah. No. In short, we don't have an automatic solution for scoring or for the detection of sleep features. But as I said also in the talk, it's rather easy to convert scores from a different software. Once you can export this to a list of scores, for example, it's rather easy to convert this to BrainVision markers as well, and then you can continue with the sleep scoring solution on those markers. This could be, for example, this could work in a semi-automatic process where you had a pre-scoring from a different software and then inspect the scores and validate them with the sleep scoring solution in Analyzer. So, but we have since Analyzer is not a dedicated sleep analysis tool, but more a general signal processing tool, we don't have an actual automatic scoring feature. However, there are also different services that provide such sleep scoring. If you're interested in that, we can recommend Physip, which is a company that applies the ASEGA algorithm to sleep data for the scoring, and they can also provide BrainVision marker files with the scores that you can directly import into Analyzer. This would be also an option for it.
Perfect, thank you. A question now for Eduardo. The long-lasting cream that you mentioned, which won't dry out for long measurement and long acquisition times, can this be used for simultaneous EEG and fMRI studies?
I would say, well, this is actually designed for active electrodes, so you can't really use active electrode technology within the scanner environment, I'm afraid. So I actually am not sure whether this is actually compatible with, well, can be safely used in the MR environment. I would actually check with the actiCHamp and actually let you know. Nevertheless, since we're talking about this kind of application, again, I would go back to the LFX. So the advantage of the LFX is that it keeps the electrodes very stable, which is, and again, as I told you, cable movement might be, an electrode movement might be sort of problematic, especially with passive electrodes. So I'm not sure what kind of results you will get with this electrolyte cream, but it's definitely a very interesting question, and I'm going to look it up and get back to you if possible. We're going to note down the email and follow up on a later stage after the event is finished. So thank you for the question.
Thank you for the answer, Eduardo. There's another question. Is your equipment licensed for clinical or medical applications?
No, as a mission of our company, we focus on research applications. We want to design products that are flexible, modular, expandable, that can be combined and integrated with different types of devices. So the answer is no. They are not. They do not possess an FDA marking or like a CE medical marking. So they cannot be used for diagnosis or treatment of medical conditions, but this is not our focus. You can go use it for clinical research, of course, but not for medical application, let's say. So
one last one has come in for Bastian, and it is about the N4 sleep stage. There is no button shown in the solution for that. Can you please explain why?
Yeah, we decided not to have a N4 staging button available because the recommendations or the guidelines by the American Association of Sleep have changed in that manner. But the solution will also recognize N4 sleep stages if you have them in marker form in the data, and then offers to convert them to N3 stages as well, as the recommendation also says. And then you can continue scoring on that. So You cannot use them in the solution itself, but it will be able to recognize them.
So with that, we will close up the webinar, and I want to thank everybody so much for attending. We hope you enjoyed and learned a lot about our hardware and software offerings for sleep research. Before leaving, we'd like to invite you to participate in the exit survey before signing off. So it just takes a few seconds for your browser to refresh, and then you'll see that. After the webinar, you'll also receive a follow-up email with a link to the PDF slides and a recording of the webinar. In this email, you'll also be invited to participate in a survey that will help us better understand your needs as sleep researchers. As Bastian said, we have a lot of content on our go-to stage where you can rewatch this and past webinars, and be sure to follow us on social media so you can stay up to date with future online events we will be hosting. So with that, I would like to again thank the speakers and thank the audience, and wish you all a wonderful rest of your day or evening, depending on where you are. Thank you.
Thank you very much.
Bye-bye.
Bye.